All right, we're going to do, let's call this nerve or neuron connections. Okay, you ready? The axon terminal of one neuron reaches out and connects with the dendrites of another neuron. And we have to look at specifically how that actually works. So the connection is called a synapse. Some people have heard pronounce it synapse. You hear both. Synapse, synapse. We're going to look at how that works. There's basically two types of synapse. There's a chemical. So there's a chemical synapse and there's an electrical synapse. So let's talk about the electrical synapse first. And now the smartboard's going to act all crazy, so let me fix that. All right, try this again. An electrical synapse. And we'll draw ourselves a little picture of how the electrical synapse works. Essentially, if you think about that long axon, how it sort of branches off at the end into a little axon terminal. What we're basically looking at is now that axon terminal will be connecting with the dendrites of another neuron like this. Okay, so this is the connection now that we want to blow up and look at in detail and see how this actually works. Okay, we'll talk after. Okay, so I'm going to draw a bigger picture, a blow up of that. So basically what we have is we have the end of that little extension. We're going to just kind of draw it like this. Right? So that represents this tiny little piece right here in the circle, blown up, looking bigger. And then it's going to sort of be, I'm going to take a different color just for clarity. And we're going to draw another membrane very, very close, essentially touching. This is the membrane of this little guy, the dendrite. So there's the connection. Okay, and then there are the membranes themselves. And remember, the membranes have phospholipid bilayers, and they have proteins and things going on, and gates and all of that. And so it's not easy for things to move from one membrane to another, but there are what we call gap junctions. So there are little channels that form sort of a connection or a pathway. All right, these are called gap junctions. So what I'm going to do is I'm going to write on here, this is the dendrite part. And over here in black is the axon terminal. And in blue here, we have these nice little gap junctions. So let me just write gap junctions. Blue things. And essentially what this means is when there's a depolarization, okay, remember from yesterday, if you have an area where your membrane has been depolarized. We'll put our little pluses here and our minuses. And then what happens is we get a little depolarization area like this traveling along here. Whoops, not listening to me. There we go. Little depolarization area. As that travels this way, it will be able to directly depolarize the dendrite through the gap junction. So this depolarization, which is caused by, remember, the movement of ions, sodium ions in particular, that are leaking into the membrane, that's happening in the domino effect as it moves along. And when it reaches the other dendrite, it's transmitted directly to the other dendrite, which causes that dendrite to have the same sort of, black. Here we go. The same sort of depolarization. I'll write the depolarization in here, right there, which then allows it to travel along the dendrite and continues on. So this is a direct transference of the, I guess you could, it is an electrical signal, but remember, it's not electricity like a zap of lightning, right? It's electricity in the sense that it's the movement of charged ions across the membrane. So when you see all these pictures on YouTube or on videos or movies about the brain, they show pictures of the neurons and they show like flashing lights as if there's electrical like shocks going through the brain. That's not really what's happening at all, right? That's a way they use to describe it figuratively so you can see what's happening, but that's not what's happening. Sticking your finger in an electrical outlet is not the same as the electrical signal of your brain. It's the movement of these ions, which is a form of electricity, but it's not quite that. So in the electrical synapse, the key thing here is that there's a direct transfer. Let me just write here, direct transfer of depolarization. That's the action potential from one nerve to another, or one neuron. Let's put neuron. Erase that, neuron. One neuron to another. The second type of synapse is a little bit different. So I'll let you catch up to me, and then we'll go on to that. Okay, some people have just pointed out that in the textbook they refer to the axon as the presynaptic, right? Presynaptic neuron or terminal, and then they refer to this one as the postsynaptic. Those are just fancy words. I mean, before and after, so there shouldn't be too confusing. Postsynaptic. We called it is the axon and the dendrite, but pre and post is basically just talking about before and after, right? Okay, let's move on to the next type of chemical synapse. Chemical synapse. It's slightly different. So let's draw another picture. Here we have the incoming axon terminal, right? Axon. Or if you want, you can call it the presynaptic terminal or the presynaptic neuron. And in the membrane, there are little, what we call vesicles, right? These vesicles contain specialized chemicals called neurotransmitters. So let's write vesicles Vesicles are just little like bubbles. They're like membrane bound. They're like kind of like a vacuole, but like a little small vesicle that is membrane bound and contains neurotransmitters, which would have been manufactured by the cell machinery. So vesicles containing N-E-U-R-O, transmitters, which are specialized chemicals that are going to do some interesting things here. So I'm going to put the neurotransmitters as tiny red dots inside the vesicles, these little chemicals. So what happens is along comes the action potential. Well, actually, you know what we could do first is let's draw the postsynaptic neuron, but we're going to draw it with a space. I'll exaggerate the space so you can see it. Here is the postsynaptic, the dendrite of the next neuron. The dendrite has very special receptors. These receptors are protein molecules or glycoproteins on the layer of the membrane that are just the right size and shape to hold a neurotransmitter. So when one of those little neurotransmitter gets there, it will fit perfectly in that spot. Remember, biology is all about shape, right? Biochemistry. So what happens is the depolarization or the action potential is flying down the axon, right? So we'll draw it here. And here we'll draw out the depolarization in red to show it. When it reaches the end here, what it does is it triggers another kind of gate to open. So you'll recall from yesterday that changing voltages are enough to open and close those protein gates. The changing voltage, if you think about everything, the shape of every molecule has to do with the pressure from electrons and charges, right, that are pushing and pulling and making everything twist and turn. Vesper theory and all those things. A voltage affects charges. It pushes and pulls on charges too. So a voltage change, like this action potential here, when it gets to a certain point in the membrane, it can change the shapes of those proteins and open or close channels, right? Yesterday we opened and closed sodium and potassium channels. Now, what happens is calcium channels open. So the depolarization, I'm going to write this underneath. The depolarization or the action potential, let's call it the action potential as it moves. The action potential causes calcium channels to open. Again, another reason why calcium is so important. We saw it with bones, we saw it with muscle. We were doing the osmoregulation of calcium, and we were doing the endocrine system and how it regulates calcium levels. Now, when these calcium channels open, this causes these vesicles to migrate to the edge of the neurotransmitter, or to the end, sorry, so they move like this to the edge. And I'm going to just use my little eraser and open them up. And when they open up, they release their neurotransmitters. Okay? Yeah. All right, so let's label these as receptor sites. Some people were a little confused. Okay, but we're not there yet, so don't worry about that. The action potential causes calcium channels to open, which, in turn, causes the vesicles to migrate to the membrane and release the neurotransmitter molecules. Okay? And are released into this space in between here. This space has a name. It's called, I'll just put it up here, it's called the Synaptic Gap, or I've noticed that some books call it a cleft. Those mean the same thing, it doesn't matter. Synaptic gap or cleft is the space in between here. You'll notice in the other one, they were very tightly together. The membranes were basically touching and had the gap junctions in between them. Here, this is a space. So the neurotransmitters, so release the neurotransmitters into the synaptic gap. So we have to be able to tell the story, right, of what's going on. Once the neurotransmitters are released, they then flow across the gap, right? So it's going to be hard for you. So on this picture, see, I can keep adding to this picture. You might want to redraw the picture, but basically we're going to put a little arrow here and show these neurotransmitters lodging into the receptor sites. Okay. What we could do to make the picture is use little points. So I could put A here in purple, and we could put an A right here to show that that's what's happening. Description A. And then over here, I'll put a B. And then down here, I'll describe what's happening at position B. That might help clear up or make it easier to use one picture. Okay? So you see what's happening there at position B. We're just going to write that underneath here. So what's happening in B, the neurotransmitters are fitting into receptor sites on the dendrite, or the post-synaptic neuron. And remember, that's because they have just the right shape, just the right chemistry to fit in those spots. And of course, when they do, they will push and pull on the protein that's attached to whatever that receptor is. And they will change the nature of the membrane. That's what these receptor sites do. That's the kind of the main mechanism of cell interaction, is molecules that can interact by lodging on receptors and cause changes in certain gates. And in this case, they can cause the sodium channels to open, and they can cause a new action potential to begin in the postsynaptic neuron. So we'll just add to that, causing a new action potential to occur. Okay, so the neurotransmitters, once they fit into their receptor sites, they alter the membrane, allow for the influx of ions, and then a new action potential can occur. So this is a little bit different because we have this chemical sort of messenger that has to bridge the gap between the two neurons at the synapse. So we'll go back to our picture for a second. Right? So we can then say that once those land in there, we can write in a new action potential right up here that's now going to travel along. It's going to travel along in the same way. So it's like a domino effect, but it has this slight little extra bit with the neurotransmitters inside. Now, there's more detail to this, of course, but we're just kind of getting the basic idea. So as an introductory kind of understanding of how this works, what you'll notice is that because we're dealing with a molecule that has to travel across the receptor sites, it is quite possible for other molecules to get in the way. Other molecules could interfere with the movement of these neurotransmitters. If you had another molecule that was a very similar shape, it might sit in the receptor site but not do anything and clog the receptor sites, which would then cause the neurotransmitter to just kind of float around in the synapse and not be able to do anything, which would essentially halt the nerve signal. But there are lots of different chemicals that can do this. We know that alcohol, when you drink it, can interfere with the synaptic function here. It can interfere with the way the neurotransmitters are being received by the postsynaptic neuron. And so what alcohol does is it either slows down or completely stops the nerve signals because it's not allowing this chemical message to get through. And that's why when you drink alcohol, a little bit of it, as it starts to work, you begin to feel a little bit uncoordinated, right? You lose your ability to coordinate your body, to make judgments, and that's because some of your neurons are being interrupted. The nerve signals are not traveling as efficiently. The more alcohol you drink, the worse it gets, and it can get really bad if the alcohol begins to block signals from nerves that are controlling your lungs. And in severe cases of alcohol poisoning, people die usually because their lungs simply stop working. They're not getting the signals they need, and they just stop working, and people end up suffocating. So too much alcohol, not a good thing. Yeah. And that's basically what happens. And there's lots of other things that can do this. We also know that there are lots of chemicals that can change the behavior of synapses in many ways. Sometimes they can enhance them. Certain drugs that we have that are, hallucinogenic drugs, psychedelic drugs that cause our brains to sort of go crazy and imagine all kinds of crazy things, are also chemicals that can mimic the action of neurotransmitters sometimes and can cause neurons to fire even though they're not being stimulated, right? So we can do the artificial stimulation of this through the synapse. All right, so what we'll do now is we're just going to look at some common neurotransmitter molecules to get an idea of what some of them are. So let's put common neurotransmitters. And we'll just make a little list and describe some of their little basic functions. I'm going to borrow from this textbook here. Acetylcholine is a very important one. So let's write it down. Just jumping around. Acetylcholine. This one is often used to excite vertebrate skeletal muscles or other different sites like that. It's secreted by the central nervous system and the peripheral nervous system. And it's usually, so I guess what I should say is, we talked about a synapse that connects two neurons, right? But eventually, when the final neuron arrives at the muscle it's going to activate or affect, then there can be a synaptic connection to the actual muscle cell. So if we go back and look at our picture, the last step on the chain, instead of a dendrite, this would be a connection to an actual muscle cell, and the neurotransmitters would move across into the muscle cell and cause the muscle action to happen. And that's one of the ones that acetylcholine, things that acetylcholine often does. So we're going to write invertebrates. And I'm just going to write neuromuscular junction. That sounds like big words, but it's not. That's grade 4 words if you think about it. Neuro means nerve, muscular means muscle, and junction means meeting point. So you should be able to understand that this is where we have the synapse connecting the nerve to the actual muscle. So we'll put a little line under that one. Other very common ones, there's norepinephrine. Norepinephrine. That one is usually the CNS and the PNS. Both secrete that one. Then we also have dopamine. And lastly, in this group, we'll have serotonin. You probably have heard of these guys. Serotonin is usually just the central nervous system, but dopamine can be both. These are our neurotransmitters that we often hear about. Norepinephrine is involved in the fight or flight, right? Activating our bodies when things get crazy. Dopamine is sometimes referred to as the happy pleasure, the pleasure chemical. When you have high levels of dopamine, that means that you're going to be able to stimulate certain nerves in the brain that create emotions and feelings. So these ones are more related to affecting the way our emotional state is. Serotonin, that's also involved in our sleep and wake cycles and things like that. So they're activating nerves, or they're connecting nerves that are involved in those processes. Then there's a bunch of amino acids that can function as neurotransmitters too. Okay, not going to cooperate today. Hang on. There we go. There's something called Gaba. And if you go on in biology next year, you're probably going to deal with a lot of these. Maybe in your probably second year, you start to get into specialized courses, going into medical school and things like that. A lot of medications that we use mimic the actions of neurotransmitters. So if people have not enough, we can give them a drug that stimulates that. Or if they have too many, we can give them another drug that helps to reduce it. Okay, we'll talk about that in a second. GABA, which stands for gamma aminobutyric acid, but we're not going to worry about that. And it's a central nervous system. It's also at the neuromuscular junction. And we're going to talk in a minute about how there's two roles that these neurotransmitters can play. They can either turn on an action potential, or they can reduce the chance of an action potential. They can inhibit an action potential. So some of these are called inhibitors. And if you get into medicine, there will be, you'll often look up medications, and they will be called things like reuptake inhibitors or things like that. So we'll talk about that in just a second once we get the list down. So just hang on, because you'll probably have seen some of these words before and go, oh, I heard of that. My sister takes this medication or whatever. Glycine is another one that we find, amino acid version. It's in the central nervous system as well, brain and spinal cord. Glutamate functions as a neurotransmitter, I think in central nervous system. That's another neuromuscular junction one. I'm just highlighting those because a lot of what we do, of course, a lot of the effectors are muscular. So a lot of the nerves that we have end up innervating a muscle, connecting with a muscle, and causing it to do something. But of course, they could also connect with glands and release other hormones, and there's other things that could happen. And then we have aspartate as the last one. Aspartate is another central nervous system one. It just turns out that we'll go back after and we'll talk about excitatory versus inhibitory. We'll get to that after. So, and then we have a couple of weird ones. Well, I call them weird ones because they're not as common. You don't hear about them when you read health articles and things like that. But this other one is called substance P. Interesting name, substance P. And what it basically is it's what we call a neuropeptide. So it's a chain of amino acids. It's a very large molecule. This one is, I'm not going to write them down, but 2, 3, 4. It's 11 different amino acids stuck together. So it's a polypeptide. It's a very large molecule. Maybe that's why it's called substance P. I'm not actually sure, because it's a polypeptide. And there's another one called metenkephalin. IN, right? Metenkephalin is actually a type of endorphin. An endorphin is a neurotransmitter that helps to inhibit or reduce pain, right? Endorphins. So substance P, this is found in both, and this one is found usually in the central nervous system. This pain, of course, is a weird. We don't really fully understand pain, to be honest, but pain is a signal that gets to the brain and is interpreted as pain, and it usually indicates some kind of injury or condition, right? Okay, so now, what I'm going to do, I'm going to come back to this list in a minute, but we're going to talk about how we can have two different sort of types or actions that neurotransmitters can take. Neurotransmitters can have two different effects on a neuron or muscle. I'm going to say on the postsynaptic cell, right? Whatever that is. Whether it's another nerve or whether it's a muscle. They can be, one, excitatory. I think it's an A. Excitatory. Or they can be inhibitors or inhibitories. So, two, inhibitory. And you can tell by those two names what they sort of do, right? An excitatory one would basically be wanting to cause some kind of action, want to cause a response to happen. Okay? Okay? All right, so back to excitatory, which would basically cause some kind of effect to happen. So let's put, causes a response to occur. And then the inhibitory one would prevent a response from occurring. Now, those are kind of vague definitions, right? Usually what happens is neurons are involved in a very complex chain or network where you have excitatory, inhibitory neurons working together to produce a very wide range of possibilities. So I'm just giving you sort of a general idea just to get the what's going on. If you think of it that way, you can basically have a good beginning. So what can happen is this. If you have a connection, so this is going to be a presynaptic nerve or neuron. And then over here, right, we have the postsynaptic muscle cell. So, if you have this neurotransmitter floating across here, when it lands on the receptors of this muscle cell, it might cause an excitatory response. It might make the muscle contract, right? Or cause something to happen. So that's going to be the excitatory. That's right, excitatory. But at the same time, on another place, on the same muscle cell, you might have another neuron that connects in a different spot, right? It's a presynaptic neuron as well. It has a neurotransmitter But this neurotransmitter is inhibitory. And so essentially you can imagine a complex network where you have many different neurons attaching to or innervating a particular muscle. Some are trying to turn it on and turn it off. And so it becomes like a little bit of a battle, right? A little bit of a, which one is more? Is there more inhibitory or is there more excitatory? And in order to get the muscle to activate, there has to be a certain threshold of excitatory action that's causing it to do its thing. And so nerves can do two things. They can activate things to make them happen, but they can also inhibit or try to stop them from happening. And this is where it gets extremely complex, because you can imagine, imagine a complex emotional state where, you're very upset at your friend, and there's a part of you that wants to instinctively lash out and punch them in the face. But there's another part of you that says, no, that's socially unacceptable and unethical, and so it holds you back. And you can imagine this conflict where you kind of feel like, you've got one set of nerves basically saying punch and another set of nerves saying don't. And it creates a very complex emotional state. Animals that don't have that capability would react far more instinctually, but humans, because of our evolution, we have the ability to have complex neural structures that involve both inhibitory and excitatory neurons that can work in conjunction and sort of create all kinds of complex situations. So there was a question. So if we go back to our list of neurotransmitters, right up here, acetylcholine, we can identify which ones tend to be more excitatory or inhibitory. So this one, acetylcholine, can do both. It depends on how it's connected and where it's going. So this one, I'll just write both here. Let's see here. Typically, though, when it's a neuromuscular junction, typically it's excitatory. If it's a junction with something else, it can be inhibitory, but acetylcholine is often an excitatory neuron or neurotransmitter that stimulates muscle action. Okay? Norepinephrine is both, depending on what it's connected to and what it's where it's coming from. Dopamine is pretty much excitatory most of the time, but I'll put a star beside it because it can act as an inhibitory neurotransmitter at certain sites. Don't forget, the more we study, the more we learn about these, and so I'm just giving you general ideas. There's always exceptions. Serotonin is generally an inhibitory It's generally an inhibitory one. So it's basically shutting things down. Okay? Gaba itself is an inhibitory one. Glycine is also typically inhibitory. Glutamate, though, and aspartate are both excitatory. Substance P is generally, is generally excitatory. And this metenkephalin is generally inhibitory. And that makes sense, because if it's an endorphin, what it's trying to do is it's trying to stop the firing of neurons that are indicating pain. So pain is coming from your toe that you just stubbed against the table leg, and it really hurts, right? But the endorphins that are being secreted are trying to block this pain response. They are inhibiting the connections to stop that nerve firing. And of course, endorphins are very important because when we are in a fight or flight situation, we get injured, we get attacked, we get hurt. We still have to be able to function. We can't be overwhelmed with pain. So the endorphins kick in, and the endorphins block pain receptors to help us to continue on. Usually it's a short-term thing, and then eventually what happens is the levels of this are going to be reduced. And that's another thing we should talk about, is reuptake of neurotransmitters. So once they're released into the synaptic gap, in order for them to fire again, there has to be something called reuptake. So neurotransmitters must also be reuptaken, if that's even a word, reuptaken by neurons after the fact. So there has to be a mechanism to clean out the synaptic gap of these neurotransmitters once they've been released. And so reuptake is another sort of whole, reuptake is another kind of, what that basically means is the neurotransmitter will be reabsorbed by the presynaptic cell, right? In other words, we're kind of taking them back after the action is over type thing. This means that there's a certain sort of refractory period or a resting period while the neurotransmitters are reabsorbed. It all happens very quickly because chemicals can do things extremely fast, right? Fractions of a second. And what can happen, and I was talking about medicines, a lot of the medicines that we have allow us to control whether or not a neurotransmitter is reuptaken. And so if you have low levels of a neurotransmitter, by preventing its reuptake, you can actually increase its levels that make them available for a synapse or a nerve connection. A lot of our mental health medications involve neurotransmitters because mental health is a very complex thing. As I said before, you can imagine all these inhibitory and excitatory neurons all acting in the brain, all these things happening. And the medications we use try to control some of that. And so sometimes you'll have what's called a reuptake inhibitor, which prevents the reuptake of a neurotransmitter, which effectively increases its availability for a synaptic connection. You can also have neurotransmitters themselves. We use molecules that are similar shaped to these neurotransmitters, or the actual neurotransmitter, and we give injections of them, or we take them orally, or whatever. We've learned that we can manipulate a lot of the action of the brain using drugs. We've also learned that we can do it illicitly with illegal drugs like morphine and or cocaine or all kinds of other drugs that can function in these ways, either as direct neurotransmitters or as a molecule that affects the release or uptake of neurotransmitters, right? All of which would affect nerve signals. And of course, in the central nervous system, if you're messing with nerve signals, you're essentially able to mess with emotional states, because emotional states are complex connections, right? So you can imagine a neural network where you have this nerve connected to that nerve, which is connected to this nerve, which is connected here, and then this connection and that connection. And all of these connections have the potential to be excitatory or inhibitory, right? And so at any given moment, there's an overall result of all of those things, which basically contributes to our mood and our emotion. Emotion is, and can be controlled. You can control emotions chemically using these neurotransmitters and all of these things, which is kind of neat. Yeah. All right, and so there we have a basic introduction to the concept of a neurotransmitter, how it works in the synapse, the fact that they can be excitatory or inhibitory, and the whole deal. So we'll stop there.